Good morning, everyone, and thank you for being here today. I'm pleased to welcome you to this presentation on a very important topic. Today, we will be discussing the regulatory and scientific justification for using 0.2 micron filters in sterile manufacturing. Over the next few slides, I will walk you through the key concepts, standards, and scientific principles that support this practice. So, let's dive into the world of sterile filtration and understand why this size is critical for ensuring product safety and compliance. Welcome to our discussion on sterile filtration, a vital step in pharmaceutical manufacturing. This process is essential for removing microorganisms and particulate contamination from heat-sensitive products that can't go through terminal sterilization. The 0.2 micron sterilizing grade filter is widely recognized as the standard in the industry. It's trusted because of its proven ability to retain microbes and because it meets regulatory requirements. Now, why is this so important? First, it ensures the sterility of the pharmaceutical products we deliver, which is crucial for patient safety. It also prevents microbial contamination, keeping the products safe and effective. Furthermore, sterile filtration supports aseptic processing operations, helping maintain a controlled environment during manufacturing. It plays a key role in protecting both patient safety and the overall quality of the product. And of course, it aligns with global regulatory expectations, ensuring that our products meet the necessary standards worldwide. All of these points highlight how sterile filtration is a cornerstone of quality and safety in pharmaceutical production, connecting directly to our overall goal of delivering safe, effective medicines. Let's take a closer look at the regulatory expectations that guide our sterile manufacturing processes. First, there are several key regulatory references we should be aware of, including the U.S. FDA's 21 CFR Part 210 and 211, which set the foundation for good manufacturing practices. In Europe, the EU GMP Annex 1 specifically addresses the manufacture of sterile medicinal products, emphasizing the importance of sterile techniques. The United States Pharmacopoeia, with chapters 1207 and 1229, provides detailed guidance on sterilization and microbial control, while PDA Technical Report number 26 offers best practices for sterilizing filtration. ISO 13408 focuses on aseptic processing, ensuring we maintain sterility throughout production, and WHO GMP guidelines, along with PIC-S guidance on sterile manufacturing, further reinforce these principles globally. Now, focusing on sterilizing grade filters, the regulatory expectation is clear. We must use filters that are validated for sterilization. This means demonstrating their microbial retention capability, that they effectively remove microbes from the product. We also need to conduct integrity testing both before and after filtration to confirm the filter's performance at each step. This is crucial to ensure the process remains robust and sterile. Additionally, a scientific justification for our filtration process is essential. We need to understand and document why a specific filtration method is suitable for our product and process. Equally important is controlling the bioburden prior to filtration, because reducing the microbial load beforehand makes the filtration process more effective and reliable. All these points together help us meet regulatory expectations and maintain the highest standards of product quality and patient safety. Now, let's take a closer look at some key applications of filtration in the pharmaceutical industry. These include injectable products, ophthalmic preparations, biologics and vaccines, cell culture media, and sterile bulk solutions. Each of these areas requires stringent sterilization to ensure safety and efficacy. Moving on, you might wonder, what exactly is a 0.2 micron filter? Well, a 0.2 micron filter is a type of membrane filter specifically designed to trap bacteria and other particulate contaminants. During sterile manufacturing processes, it acts as a critical barrier, preventing unwanted microorganisms from passing through and contaminating the final product. When it comes to the materials used for these filters, there are several common options. Polyethersulfone, or PES, is known for its excellent flow rates and chemical resistance. Polyvinylidene fluoride, or PVDF, offers high purity and durability. Nylon filters are versatile and widely used, while PTFE, or polytetrafluoroethylene, provides exceptional chemical resistance and is suitable for filtering aggressive solvents. Understanding these materials and their applications helps us appreciate how vital proper filtration is in maintaining the integrity and safety of pharmaceutical products. Let's take a closer look at the scientific basis for the 0.2 micron filtration. The core principle here is microbial retention, which involves several key mechanisms. First, size exclusion plays a crucial role. Microorganisms are physically blocked because they are larger than the pore size of the filter. Then, there's adsorption, where microbes stick to the surface of the membrane, adding an extra layer of protection. Additionally, the tortuous pathways within the membrane create a complex route that microbes cannot easily navigate through. Now, the scientific justification for using a 0.2 micron filter is based on the fact that most bacteria relevant to pharmaceutical contamination are actually larger than 0.2 microns. This means that a properly functioning filter can reliably retain these microorganisms. When it comes to validation, we use a standard challenge organism, which is approximately 0.3 microns by 0.6 to 0.8 microns in size. This organism is used to test and validate that our sterilizing grade filters are performing effectively. The main objective here is to demonstrate that the filter can consistently retain microbes, even under the worst-case conditions. In other words, we want to be confident that the filtration process is reliable and robust, ensuring product safety and compliance. Let's talk about why 0.2 micron filters have become the industry standard. First, they have strong regulatory and scientific acceptance, which means they are trusted by experts worldwide. Their proven bacterial retention capability is a key factor. These filters effectively remove bacteria from liquids, ensuring safety and quality. They're widely validated and accepted by regulators, so you can be confident in their performance across various applications. They're especially effective for sterilizing filtration of liquids, which is critical in many industries like pharmaceuticals and biotech. Another important point is their compatibility with aseptic processing requirements. This means they support sterile environments without compromising the process. Plus, they help maintain the integrity of heat-sensitive products, so you don't have to worry about damaging delicate formulations. Now, from a regulatory standpoint, 0.2 micron filters are recognized as sterilizing-grade filters, as long as they are properly validated. This validation is essential to confirm they perform as intended and meet industry standards. So overall, choosing a 0.2 micron filter gives you a reliable, scientifically backed solution that supports safe, effective, and compliant sterilization processes across a wide range of applications. Now, let's talk about sterilizing filter validation. When we validate these filters, there are several key studies we need to include to ensure everything is up to standard. First, bacterial retention testing is crucial. This shows us that the filter can effectively remove bacteria from the product. Next, product compatibility studies help us confirm that the filter doesn't negatively interact with or damage the product itself. We also conduct extractables and leachables evaluations, which check for any substances that might come off the filter and contaminate the product. Chemical compatibility testing is similarly important. It confirms that the filter materials won't react with the product over time. Flow rate and pressure studies are performed to ensure the filtration process is efficient and consistent under normal operating conditions. Lastly, adsorption and product binding assessments help us understand if the filter might adsorb or bind to any part of the product, potentially reducing its efficacy. Now, from a regulatory perspective, the main expectation is that validation must prove that the filtration process reliably produces a sterile filtrate, meaning no microorganisms are passing through. and that this process doesn't compromise the quality of the product. This is fundamental to ensuring patient safety and maintaining compliance, tying all these validation activities together within the broader scope of quality assurance. Now, let's take a closer look at bacterial retention testing and the validation standards we follow. In this testing, filters are challenged with high concentrations of Brevandemonas diminuta, which is a standard challenge organism because of its small size and relevance to sterilization validation. The typical challenge conditions involve exposing the filter to a minimum bacterial concentration of 10 to the 7th power CFU per square centimeter, representing a very rigorous test scenario. We also simulate worst-case process conditions and maximum filtration area challenges to ensure the filter's robustness under the most demanding circumstances. The acceptance criteria are straightforward. No bacterial passage through the filter is allowed. This is a critical point because it provides scientific evidence of the sterilizing capability of the filter. In other words, if the filter passes this challenge, we can be confident that it effectively retains bacteria even under extreme conditions. This testing and validation process is essential for maintaining high standards of sterilization and ensuring product safety. Now, let's talk about the integrity verification of sterilizing filters. When we perform integrity tests, there are a few common ones to keep in mind. The first is the bubble point test, which essentially checks the pore size and whether the filter can hold pressure without leaking. Then there's the diffusion test, which measures how well the filter prevents particles or microbes from passing through by analyzing the diffusion of a gas or liquid. The third is the pressure hold test, where we apply pressure and monitor whether the filter maintains its integrity over time. Moving on to regulatory expectations, it's important to note that the EU GMP Annex 1 recommends conducting pre-use and post-use integrity testing, often called PUPSIT, to ensure ongoing filter performance. And critically, these integrity tests should always correlate with the filter's microbial retention capability. That way, we can be confident that the filter isn't just intact visually or physically, but also effective at keeping microorganisms out. The main objective here is to ensure that the filter remains both intact and functional throughout manufacturing. This is vital because any compromise could lead to contamination and impact product safety. So, thorough and regular integrity testing isn't just a regulatory requirement. It's a key part of maintaining quality and safety in our processes. Let's talk about bioburden and prefiltration control. First, why is bioburden control so important? Well, a high microbial load can really cause issues. It can overload the filter, making it harder for it to do its job effectively. This not only reduces filtration efficiency, but also increases the risk of contamination, which is something we definitely want to avoid. So, what measures can we take to control bioburden? Creating a controlled manufacturing environment is key. Using prefiltration systems helps to catch contaminants early. It's also crucial to process materials in a timely manner to prevent microbial growth, and ensuring that all equipment and pipelines are properly sanitized helps maintain a clean process overall. From a regulatory perspective, the focus is clear. Bioburden should be minimized before the sterilizing filtration step. This is essential to ensure product safety and compliance. Keeping bioburden low not only protects the integrity of the filtration process, but also supports the overall quality of our final product. Now, let's take a closer look at the applications of this technology in sterile manufacturing. In pharmaceutical manufacturing, it plays a crucial role in producing various sterile products. For example, it's used in parenteral preparations, which are injections directly into the body, requiring the highest level of sterility. It's also vital for making vaccines and biologics, which are often sensitive and need to be free from contamination. Ophthalmic solutions, like eye drops, also benefit from this technology to ensure purity and safety. Additionally, it's used in the production of intravenous fluids and sterile active pharmaceutical ingredients, or APIs, which are the raw materials for many drugs. Media filtration is another key application, helping to remove microorganisms from liquids. When we look at manufacturing areas where this technology is applied, we find aseptic filling lines, which are specialized stations for filling sterile products into containers without introducing contaminants. Buffer preparation systems are also involved, where ingredients are prepared under controlled conditions. Final bulk filtration operations are the last step to ensure that the finished product is sterile before packaging and distribution. Now, what are the main benefits? Primarily, this technology supports sterility assurance without the need for heat sterilization. That's a significant advantage because it preserves the integrity of sensitive products that could be damaged by heat. Overall, this approach helps us produce safe, effective sterile medicines while maintaining the quality of the final product. On this slide, we're focusing on some common regulatory observations that we often see during inspections. These are areas where deficiencies tend to frequently occur and can have serious implications. For example, inadequate filter validation documentation is a major concern because it directly impacts the sterility and safety of the product. Missing integrity testing records can also raise red flags as they prevent us from verifying that filters are functioning properly. Another critical point is the lack of product compatibility studies, which are essential to ensure that the filters and materials used won't adversely affect the product quality. Inadequate bioburden monitoring is also a common issue, as it can lead to contamination risks going unnoticed. Poor change control management can cause inconsistencies and traceability problems, and improper filter installation or handling can compromise the entire sterilization process. Now, let's discuss the potential consequences if these issues are not addressed. The most immediate concern is sterility assurance, because any lapse can lead to contaminated products reaching patients. This can result in product recalls, which are costly and damaging to the company's reputation. Furthermore, regulatory agencies like the FDA may issue warning letters or observation notices, which can lead to further scrutiny and penalties. In summary, these common deficiencies highlight the importance of rigorous validation, documentation, and control processes in maintaining compliance and ensuring patient safety. Keeping these points in mind helps us understand why thorough regulatory adherence is so critical throughout our manufacturing and quality assurance activities. Now, let's talk about risk management and change control, which are crucial for maintaining filter performance throughout the product lifecycle. First, when it comes to maintaining filter effectiveness, we need to be aware of potential issues like filter blockage or fouling, which can happen over time and reduce efficiency. Product adsorption is another key factor, as some materials may stick to the filter, affecting performance and product quality. Pressure fluctuations can also pose challenges, indicating possible blockages or system issues that need attention. Additionally, chemical incompatibility between filter materials and process chemicals can cause degradation or failure, so selecting the right materials is essential. Moving on to change control requirements, any alterations in the process require proper assessment. For example, if we change the filter material, we need to evaluate how it impacts performance. The same applies to process parameter changes. Adjustments in flow rates or pressures need careful review. Changes in the product formulation may alter filtration needs, and even supplier changes require thorough evaluation to ensure quality and consistency are maintained. The ultimate goal here is clear. We want to maintain validated filtration performance throughout the entire product lifecycle. This means proactively managing risks and implementing effective change control to ensure our filters perform reliably, safeguarding product quality and patient safety at every stage. Let's now look at some key strategies for strengthening sterile filtration systems. First, it's essential to use validated sterilizing grade filters. This ensures the filters perform reliably and achieve the necessary microbial reduction. Routine integrity testing is equally important. It helps us verify ongoing filter performance and catch any issues early. Conducting periodic revalidation is a good practice to confirm that our filtration processes remain effective over time, especially after any changes or maintenance. Maintaining robust bioburden control is critical to prevent contamination from the start. Training personnel thoroughly on aseptic filtration practices is also vital. People are often our first line of defense against contamination. Implementing a risk-based contamination control strategy, or CCS, allows us to focus our efforts where they're most needed, making our processes more efficient and safer. And of course, ensuring complete documentation and traceability is fundamental. This way, we can track every step, verify compliance, and facilitate investigations if any issues arise. Moving to continuous improvement, it's important to trend filtration failures and deviations. This helps us identify patterns and areas for enhancement. Monitoring filter performance metrics provides ongoing insights into system health. Lastly, reviewing emerging regulatory expectations keeps us aligned with industry standards and ensures our practices meet or exceed current requirements. All these points connect back to our overall goal, maintaining the highest quality and safety in our sterile processes. Let's wrap up with the key takeaways from this presentation. First, 0.2 micron filters are widely recognized around the world as sterilizing-grade filters, which are essential for aseptic manufacturing. Their ability to effectively remove microbes is backed by scientific validation, giving us confidence in their microbial retention capabilities. Regulatory bodies also require that our filtration processes be validated and that we perform integrity testing to ensure ongoing reliability. This means we need to consistently verify that our filters are functioning properly to maintain sterility. Additionally, proper bioburden control and risk management are crucial. They serve to strengthen the overall sterility assurance, minimizing contamination risks. Ultimately, effective filtration systems are vital because they protect product quality, ensure patient safety, and help us stay compliant with regulations. When we prioritize these aspects, we support the integrity of our manufacturing processes and the trust in our products. For further support, please feel free to contact us. We're here to help you with any questions or additional information you might need. And finally, I want to sincerely thank you all for your time and attention today. Your engagement is what makes this worthwhile, and I hope you found this presentation valuable. Remember, whether it's follow-up questions or guidance, don't hesitate to reach out. Thank you again, and have a great day.